Okay, welcome to my class, and today we are going to learn the last but the most important section of the diseases of the breast. I think you might guess it's the breast cancer. Breast cancer is a global health problem with more than 1 million cases of breast cancer diagnosed worldwide each year. In China, breast cancer is the most common neoplasm of women. The overall incidence of breast cancer was rising because of the increases in the average lifespan, the lifestyle changes, because we have birth control and fewer child. That increases the risk for breast cancer. The survival rates in women with breast cancer have steadily improved over the last several decades. The decreased mortality from breast cancer is thought to be the result of early detection via mammographic screening and improvements in the therapy. The pathology of the breast cancer generally divided into two main parts: the non-invasive breast cancer, also called the carcinoma in situ. The other is invasive breast cancer. Non-invasive neoplasms are broadly divided into two major types: the lobular carcinoma in situ and the ductal carcinoma in situ, which is short for LCIS and DCIS. LCIS was initially believed to be a malignant lesion, but is now regarded more as a risk factor for the development of breast cancer. As for DCIS, it is recognized as discrete spaces filled with malignant cells, usually with a recognizable basal cell layer made up of presumably normal myoepithelial cells. DCIS frequently coexists with otherwise invasive cancers, and they are more dangerous, and form microcalcifications which could be detected by mammography. Invasive breast cancer is divided into lobular and ductal histologic types, and also have some specified invasive breast cancers. And invasive ductal cancer, also known as infiltrating ductal carcinoma, is the most common form of breast cancer. It accounts for over 50%, even to 70% of invasive breast cancer. And invasive lobular carcinoma accounts for up to 10% of breast cancer. And breast cancer is highly heterogeneous. They might have the mixed part, and the different histological types will mix together. And for some specified invasive breast cancer, the medullary carcinoma is the most common one. Here is some pathologic sections of the breast cancer. The picture A showed a typical invasive ductal carcinoma. The tumor cell infiltrated stroma and formed the ductal-like structure. Depends on the differentiation of the cells. A picture B showed the invasive lobular carcinoma. We can see the single-filed tumor cells which formed concentric cycle, targetoid pattern. And picture C showed the special type of breast cancer. We call that mucinous or colloid carcinoma. We can see the tumor cells floating in the lake of muci. And the picture D showed the invasive tubular carcinoma, which is very similar to the normal breast. But the cancer invades as small tubules lined by a single layer of well-differentiated cells. And the last picture is the medullary carcinoma. The cells are poorly differentiated and with pleomorphic nuclei. The distinctive feature of this tumor are the infiltrative of lymphocytes and the syncytium appearing of tumor cells. In general, the special types of breast cancer always have better prognosis compared to the IDC or LLC. There are numerous pathways and molecular markers that have been reported to affect breast cancer outcomes, including the steroid hormone receptor, for example, estrogen receptor, progesterone receptor, and the human epidermal growth factor receptor pathway, we short for HER2, and angiogenesis factor, the cell cycles, the CDKs and P53, etc. The microarrays representation of human breast cancer indicates the different gene expression of ER-positive, HER2 amplified, and basal-like breast cancer. The molecular markers are used to select patients for systemic treatment, for example, the chemotherapy, endocrine therapy, and to predict the response of patient to the treatment. The simplest example is the use of ER or HER2 status to predict the response to endocrine treatment or trastuzumab treatment. Trastuzumab is a HER2 inhibitor. And Ki-67 is also a very important marker because it indicates the mitosis activity of cells. The higher this marker is, the more sensitive of the tumor to the chemotherapy. So in the clinical, we call that immunohistochemistry 4, because they could simply divided breast cancer into 4 main subtypes: the lobular, lobular A, lobular 2, the HER2 overexpression, and the basal-like triple-negative breast cancer, which very, very important to choose of the proper treatment of breast cancer.